What is that? Yeah, here I'm. All right, let's start. Good afternoon, everyone. Welcome to the Fellows Research Talk Session 2, focusing on neuroscience. My name is Debabrata Das, research instructor at the Department of Genetics, MD Anderson Cancer Center. I will be moderating this session along with my colleague, Dr. Sani Narayanan, research faculty at Florida State University. In this session, we will have 5 outstanding research talks. Just as a reminder, each presentation will be 15 minutes followed by 5 minutes Q&A. Fellows will be notified via private message when there are 2 minutes remaining, and the moderators will give a verbal remark if 15 minutes is over. At any point of time, please post your question in the Q&A box on your screen. Since time is limited, maybe we will not get to everyone's question and may paraphrase or combine some of the questions. So without further delay, let us start with our first speaker, Dr. Jennifer Bussell. Jennifer received her PhD from Rockefeller University and is currently a postdoctoral researcher in the laboratory of Dr. Richard Axel at Columbia University's Zuckerman Institute for Mind, Brain, and Behavior. She studies mechanisms behind curiosity and information seeking and is interested in understanding how the value of knowledge guides behavior. She is the recipient of a junior fellowship from the Siemens Foundation Society of Fellows and a Women and Science Fellowship. With that, I would like to request Jennifer to take over the stage.
Okay, thank you so much. All right, there are my slides. Are we okay? You can see them? Okay. So I'm going to talk to you today about the desire for knowledge. I'm going to show you that mice seek information as if it is its own reward. And I have identified representations of the intrinsic value of knowledge in the mouse brain. And through this work, I've developed a model system in the mouse to study the neural circuit basis of this incredibly important and yet elusive desire for knowledge. So humans and animals seek information. We constantly engage in actions to gain knowledge about the world and improve our decisions to maximize rewards. We are also curious. Like Dr. Hrabowski said this morning, we seek information not only when it is useful, but we also seem to value knowledge in and of itself. In other words, information is its own reward. And remarkably, this fact, seeking information for its own sake, is true of both humans and many other species when we test them in the lab, including monkeys, pigeons, and rats. And moreover, both functional MRI studies of humans and single-cell electrophysiology in monkeys have shown that the brain's reward system responds to information as if it is a reward and has intrinsic value. What is less clear is how potential sources of information are assigned value. How do we know how valuable a piece of information might be? When we're curious, how do we know what to explore? In other words, how is the unique value of information computed and represented in the brain? So to answer this question, I've turned to the mouse as a new model system in this field. And in mice, not only do we have access to incredible tools, we know a great deal about how olfactory stimuli in particular are represented in the brain. So I've asked how the neural representations of neutral odors, are transformed when the mouse learns that they predict information. So I've developed a task inspired by those used to study information seeking in monkeys. Thirsty mice begin a trial by poking in a center port. There they receive one of 3 odors on each trial. Either an odor that directs them to the left, an odor that directs them to the right, or an odor that offers them free choice between left or right. And for each mouse, one of the side ports provides information. This is counterbalanced across animals. So for this mouse, if it goes to the left, it's chosen the information port. There it receives one of 2 odors. Odor A is always followed by water reward. Odor B is never followed by water reward. If the mouse goes to the right, it receives a control odor, C or D, which has no relationship to the water reward. The water reward amount of 16 microliters at 25% probability is the same on the 2 sides. So here's the full task. Again, mice initiate trials by poking in the center port. When the mouse chooses the information side port, the odors there reveal immediately whether they will receive water on that trial. On both sides, there's a 10-second delay before the water reward outcome. And so choosing the information side does not allow the mouse to get additional water reward. It takes mice 6 to 8 weeks to learn this task through the training curriculum I've developed. However, once they've learned the task, they understand that one of these odors in the center port predicts information, while the other predicts an equal amount of water value, but no information. So now I'll show you that the behavior of mice trained in this task suggests that mice seek information that offers them no additional external water value. We measured the percentage of the free choice trials in which mice choose the information side across several sessions, in which we reverse the location from information from the left to the right, or vice versa within each animal. So each of these bars shows the mean preference of an individual animal, and across the population, mice showed a strong preference for the information port. And this preference for the information port suggests that mice value the information provided there. We asked whether mice were willing to exchange water value to get that information. That is, will mice pay for information? And this question of paying for information is a classical canonical economic test of whether a species values information in its decision-making. And indeed, we found that mice were willing to sacrifice up to 6 microliters of water per trial to receive this information. So together, these behavioral experiments showed that mice assigned subjective value to information, separate from the value of the external water reward. This suggests that there might be a neural representation of information involved in their decision, and that we could ask whether that information looked like value. So to do that, we imaged neural activity using miniaturized microscopes in orbitofrontal cortex in the mice. Orbitofrontal cortex, or so orbitofrontal cortex, or OFC, is thought to represent, among other things, value during decision-making, and it receives direct projections from primary olfactory cortex. So we wanted to know if OFC encodes a representation for the value of information in response to odors in this task. So we looked at responses to the center port odors, which once the animal has learned the task, predict information. And the responses I'll show you to those odors are balanced across the prediction of information on the left and the right side, because we registered the identity of cells across reversal sessions. And we did this because we, like others, observe extensive encoding of the side choice direction in OFC in this freely moving rodent task. So looking at the activity in response to these odors with information balanced across the left and right side, let us disambiguate responses to information. So here are the responses across all of the cells we observe in OFC in response to the information predicting center port odor. We see robust responses to this odor in nearly 40% of the cells in OFC. Here are the responses to the no information predicting odor in those same cells, the cells are sorted across the heat maps. You can read a single cell from left to the right. They're sorted by the difference in their response to these two odors. And so 20% of the OFC population shows a differential response to the information and no information center port odor. I'm showing you this differential response here as the absolute value of the mean difference in activity across cells in the population. And this 20% of cells contribute to a differential representation of information along an axis of activity we computed across the population on each trial in a principal component space. And moreover, we can readily decode the animal's choice to receive information, both on the directed trials and on free choice trials from this activity. So this differential activity in response to the odors that predict upcoming information, because it also predicts the animal's choice to receive information, which our behavior suggests that they value, we think of this activity as a representation predicting the value of information. But we wanted to understand if this information prediction representation scaled with values such that it might encode the amount of information value signaled by an odor. Or in other words, is the information prediction representation a scalar value signal? And so to ask this, we decreased the delay between the receipt of information and the reward outcome on all of the trials. So in psychological theories of information seeking, and it's been shown in experiments in humans and other animals, this decrease in delay should decrease the value of information because it diminishes the duration of uncertainty the animal experiences if they don't choose to get the information. And so we decreased the delay on all trials between the information and the reward on blocks of several sessions, and we fit a psychometric curve. So here I'm plotting the length of the delay versus information preference. And we observed that mice became indifferent to information at a delay of between 1 to 4 seconds. And we modeled the trial by trial choices of mice across these sessions. And in our model, we used one reward reinforcement learning value term for water reward and a separate reinforcement learning term for the value of information, where we fit a parameter for each animal for their sensitivity of information value to the delay. And so only by including both of these separate value computations for water reward and information were we able to match the preferences of mice for trade-offs between water amount and information with these various durations of uncertainty. So then we looked at the neural population responses in OFC of this information prediction representation. And I'm showing you that here as the mean absolute value of the difference between the odor that predicts information and the odor that predicts no information. And so if we look at sessions that are just about a week apart in time and both have a 10-second delay, we see almost no difference in this representation. But if we compare sessions with a 1 second and a 10-second delay, the magnitude of the information prediction representation is decreased in sessions with a 1 second delay. And these data suggest that the information prediction representation is a representation of the value of information, which at least is in part determined by the duration of uncertainty in the task. So I didn't have time to show you our data, but we found that the neural ensembles that contribute to the information value representation are somewhat overlapping but distinct from those that represent predicted water value. And this is consistent with single unit recordings in monkey OFC, in which individual neurons responded in an uncorrelated way to visual stimuli predicting different water amounts and information. And so these data and our cognitive decision model suggest that OFC maintains unique representations of the intrinsic value of information and the extrinsic value of water during this decision-making task. So we wanted to examine this at the population level. And we created a nonlinear latent variable model of OFC activity during the task. The specific modeling approach we used, Zebra, is new, and I would love to talk to folks offline about its advantages. But what I'd like to show you here are the projections within this low dimensional manifold of OFC activity in our model of the activity related to information and water reward. So these are the trajectories of OFC activity in low dimensional space on the information and no information trials, and these diverge after the center port odor. The reward-related activity, so water versus no water, diverges after the side odor that reflects information, and again at the reward outcome. And we also saw the same scaling of the distance and the difference between information and no information with diminished delays in the model that we saw in our raw neural data. And so the fact that we can observe both of these differential activities in the zebra model implies that these two kinds of value, intrinsic information and extrinsic water reward, are represented in OFC low dimensional dynamics during the task and potentially decodable from that population activity by downstream areas. So I've shown you that mice value information in their behavioral decisions. Neurons in OFC represent the possibility of gaining information. I didn't have time to show you the data, but we saw that the representation of information is distinct from, in fact, in different neural populations than the representation of water reward value. And this information representation scales with the value of information as determined by the duration of uncertainty in the task. So these results from my postdoc work suggest a possible circuit mechanism for recognizing that a sensory stimulus provides information and assigning it value. And this computation is a crucial missing gap in our understanding of the desire for information from neural studies in humans and non-human primates. So the question is, how does the brain know that a sensory cue is information that has value? And unlike other rewards like water, whether something is information depends on the organism's knowledge, and assigning it value is a process of metacognition. Nevertheless, my data and these findings provide a path forward which my future lab will explore. So in the mouse olfactory system, piriform cortex represents odor identity. With learning, representations of the water value of an odor emerge in orbitofrontal cortex. I hypothesized that the information value representations we see arise similarly. So I didn't have time to tell you, but the information prediction representation we see emerges with learning in the task. And we also see a representation of information receipt, a representation of information as an unconditioned stimulus in OFC in response to both of the odors at the side port that provide information. So this implies that there are ensembles of neurons in cognitive areas that recognize odor as an information unconditioned stimuli, and that identify stimuli as information and intrinsically valuable, and could potentially elicit reinforcement that leads to the emergence of the OFC representations of the information predicting odor. And so my lab will investigate this circuit with the tools of mouse systems neuroscience. We will examine candidate cognitive areas that are outputs from piriform cortex, looking for activity that represents information as an unconditioned stimulus of value. And we'll study this activity as interacting populations of neurons within the circuit to understand the computation of information value. And in the long term, my vision for our work is to expand into studying the role of information value representations in more naturalistic, curiosity-driven behavior in the mouse, to understand how information value motivates us to learn. And with that, I'd like to thank my mentors, Richard Axel, Larry Abbott, my collaborators, and the research assistants that have performed all of our extensive mouse training, and our funders, and I'll take any questions. Thank you.
All right. Thank you, Jen, for this wonderful talk. Let's see, we have some question in the Q&A. Sunny.
Yes, thank you, Dr. Bissell, for your wonderful presentation. Really insightful. There currently do not appear any questions in the from our audience, but I have a question. With regards to your research, how does the age of an organism affect their ability to sense information? Is there a difference in processing young versus old?
Yeah, so one of the reasons I was so excited about doing this work in mice is that we're able to kind of get at things in larger populations in a different scale than have been studied before in this process. But unfortunately, because it takes us so long to train the task, we actually aren't able to ask about like really young animals and maybe, if they have a more desire for information. We do know that, in human beings, our sort of level of curiosity and interest and engagement with the world does change over the lifetime. And that is something I'm more interested in exploring in the long term. But also mice are able to smell sort of throughout their lives from the beginning. So in terms of the sensory angle, I think it's probably pretty similar.
Thank you for sharing that. We have a question from our audience being, what is the simplest organism you believe possessed the characteristics of being able to respond to information? Where did that originally evolve from?
Yeah, I mean, that's a good question, right? There's, I think, some evidence. There's an interesting study from a guy named Adam Calhoun looking at, C. elegans, and they're sort of leaving an exploratory behavior. And, certainly almost anything that can move needs to kind of navigate its environment. Whether there's an awareness of that is, I think, maybe sort of a philosophical question. Like, I don't know if I'm confident about making statements about like whether bacteria are curious, right? But certainly almost everything that has been looked at does have this tendency and need to learn about its world. And we think, I mean, the way that I've most recently been thinking about this is that, mostly it's useful to know things, and we seek out knowledge because it helps us make a better model of the world. And so there may be this evolved, almost heuristic bonus to getting new information that has evolved and kind of assigns that as a default that's then evolved to more elaborate curiosity like behaviors in, say, humans. Sorry, I can't see everyone.
Yes, we had another question being, our next question being, if you were to switch the meaning of the odors, for example, going from an information prediction odor to an odor that doesn't predict information, would you see the information coding extinguish, or do the representations compete with each other somehow?
Yeah, so the way we've actually done the experiments is we're already looking at the information representation across multiple odors. So because we have this issue of the mice actually choosing to go to the left or the right, the information, that representation that we're looking at is kind of agnostic to the actual odor identity that we're signaling. So half of the responses to information are, cinnamon telling the mouse go to the left, and the other half in the same mouse and the neural responses we're looking at are vanilla telling the mouse to go to the right, but they both are signaling information. And so we think this is representation we've identified is really about predicting that the mouse is going to get information, not a response to a specific odor or a specific direction. I hope that was the question. Maybe they can clarify if that wasn't what they meant.
So thank you for sharing that. Another question, likely our last for this session at least. The question being, if water was restricted with the mice to motivate them to complete a task. The question being, would there still be a drive to gain information in the absence of this restriction, water restriction?
Yeah, that's an excellent question, right? So a lot of what we know about neural responses to information, what's been studied in this system is sort of these constrained types of tasks where you offer animals or subjects the choice to receive information about upcoming events, which is, sort of narrow. And one of the major things I want to do in my lab is ask about desires for information that are not about predicting something else that's salient to the animal. So even if we can just ask, if a mouse learns to associate odor A with odor B, because odor A always predicts odor B, do they want to know that? And is there some kind of neural representation of that being a reward that is more generally motivating us to learn in our environment? And yeah, for sure, that's I think probably that's true, but it's much less well studied and understood.
Thank you, Dr. Purcell, and we look forward to learning more and hearing more about your future research projects.
All right, thanks.
All right. So our second speaker is Dr. Ismail Ahmed. Ismail is currently a postdoctoral fellow at New York University Grossman School of Medicine in the laboratory of Dr. Robert Frumke. He is interested in understanding the influence of neuropeptide on complex social behaviors in diseased states and in developing innovative tools to gain a deeper understanding of how neuropeptide oxytocin modulates maternal behavior. His research is supported by the NIH and the Burroughs Wellcome Fund. Ismail, the floor is all yours.
All right, thank you all for having me. It's a pleasure to join this group of fellows and share my research with all of you this afternoon. Today I'll talk about development of some chemical tools to study neuropeptide signaling and manipulating neuropeptide signaling. Just for a little bit of background, I'm a chemist by training, and I'm bringing that to the realm of neuroscience to better understand molecular control of neural signals. All right, so just a brief overview. Communication in the brain for about 100 years. Now it's believed that one neuron talks to another neuron through synaptic inputs. A lot of this is electrical and chemical communication through small molecules, monoamines. And through those communications, you have many different behaviors. However, only recently, a new class of chemical molecules, neuropeptides, which there are now known to be over 100 of them, add a huge diversity and richness to the dynamics of circuits. And so the neuropeptide that I'm going to share with you today is oxytocin. So just a brief overview on how about oxytocin is that oxytocin is a 9 amino acid peptide. It's synthesized primarily in the hypothalamus, and it was first discovered in the early 1900s during childbirth and lactation. It has a single primary receptor, and I say single, this becomes a little important later in the talk, and it's primarily a GPCR second messenger system. Oxytocin is important because it's evolutionarily conserved and it's widespread in the animal kingdom. It goes way back to the flatworm, and it's found across many taxa. Also, it's been implicated in many different social behaviors, from mating, paternal behavior, aggression, both attack and defensive behaviors, empathy, cooperation, social recognition. And our favorite one in the Frumke lab is maternal behavior, which is crucial for survival of species, and it's the first social interaction any of us probably have had in our lives. Also, I want to mention that oxytocin is important for medicinal therapy. It's being explored for pain, obesity, and various neuropsychiatric disorders. Just to give you a view of how extensive the oxytocin signaling system occurs in the brain, oxytocin is primarily synthesized in the PVN and SON and has many outputs throughout many different regions of the brain. There's also many different inputs into the PVN to elicit release of oxytocin. On top of that, oxytocin is released in mass into the periphery, and this is also important for later in the talk. So what I wanted to ask myself is, what is the strategies for controlling oxytocin signaling, right? So primarily in neuroscience, we use optogenetics. So you get spatial temporal control with light, and you can genetically encode which neurons you want to activate. And then you use pharmacology, where you have control of systemic concentration, and you have molecular control of the endogenous ligand or receptor that's actually controlling this system. So I asked myself, how can we probe the role of specific receptors or signaling pathways in control of physiology and behavior? So can we merge the two pros of these techniques, optopharmacology, and there is this field called optopharmacology. So what I did here is I have, let's say, for example, in this large circle, let's say that's the oxytocin molecule, and chemically I add a cage to the peptide so that now it's unable to bind the receptor. And in the presence of light, in this case UV light, the cage comes off through photolysis, and that exposes now the peptide, its ability to bind the receptor. And then that elicits a downstream second messenger signaling pathway. And what I really like about this is that you have controlling over the timing, the location, and the dosing. And some key features that I think are really useful for the field is that you have control over neurons and non-neuronal cells. You can control a single molecule or receptor of choice that's endogenous to the system, and you do not need genetics. And this is useful if you're studying, for example, non-model organisms where you don't have access to the genetics. And this is increasingly becoming very popular because a lot of different social behaviors are being studied in many different non-model organisms these days. All right, so where do we start for designing a cage oxytocin? There are many different functional groups on the peptide that you can modify with adding a cage, so you would have to make many different molecules. At the time, there was no structure of the peptide bound to the receptor. So we went into the literature to look at which amino acids, if you mutate them, affected the binding most. And what it turns out to be is this tyrosine, this disulfide, and this N-terminal NH2. So I wanted to start there. So basically, I added a photo cage to those different regions of the peptide, and then did in silico binding. So this is halfway through the project, a structure came out, a cryo-EM structure of oxytocin binds to the receptor. So I was able to do in silico screening to look at what the binding would look like and see which ones would diminish the binding the most. And what through the predictions, the worst binder would be the modification of the tyrosine. So this one in red. And so then I did I tested this in a calcium flux assay in cell culture. So in buffer is in white, very low fluorescence. And this is CHO1 cells loaded with a calcium sensitive dye. So upon binding of oxytocin, you have an increase in fluorescence. But then when you have this cage oxytocin, it's the modification at the tyrosine cage oxytocin 1. It's similar to that of buffer, but then only when you add in the LED, you recover that. And so the clear winner here was the modification of the tyrosine. And that's where we move forward with the rest of the study. So this is just to show what that looks like in terms of calcium imaging. So before uncaging, so this is with the compound in solution, but before uncaging with the light, you have very low fluorescence. And then after uncaging, you have an increase in fluorescence through the release of calcium. And this is what it looks like in real time after uncaging. All right, and to quantify that binding, we show that using a competitive binding assay that is 280 times weaker with the cage on it. And then after uncaging, it recovers to that of oxytocin because it's just removing the caging group. So then I wanted to validate this in vivo or ex vivo slice. And so we did this in the auditory cortex. So this is oxytocin receptor neurons expressing TD tomato in the auditory cortex. So we patch, and so here this is a wash on of oxytocin. You have depolarization in oxytocin receptor positive cells, and then flatlined on the oxytocin receptor negative cells. And then here we have the similar results with uncaging with 365 nanometer light. You have this depolarization in oxytocin receptor positive cells, and then it's flatlined in oxytocin receptor negative cells. So then now we wanted to take this to the realm of in vivo, which has never really been done before with a neuropeptide, and only one example with a neurotransmitter. And now there's a cage drug that's also been used. This is the first with a neuropeptide. But to give you a little background, the behavior that I'm looking at is pup retrieval behavior. So Bianca Jones-Marlin, who used to be in the Franke lab, who now has her own lab at the Zuckerman Institute at Columbia, she did this seminal work where she showed, for example, that when pups are isolated from their nest, they will emit a distress call, and the damned experienced mother will go and retrieve that pup. However, a naive virgin who has no experience with pups will ignore the pup. And so what she did was she supplemented oxytocin IP and optostimulated PVN neurons in the auditory cortex and showed that there was an increase of retrieval behavior upon co-housing. However, saline was diminished, and the dams, of course, retrieved 100% of the time. In doing these experiments, we didn't exactly know where the oxytocin was acting. So we wanted to be able to do that with this tool and recapitulate these results. All right, and so this is just what den behavior looks like for retrieving. So the pup is emitting a distress call. You won't be able to hear it. It's very, you need a $10,000 mic to do that. And it brings it back to the nest. So this is just the behavior setup. So just so that we don't, so it's all done in the dark with red light so that we don't uncage ambiently. A microphone to pick up the pup calls, a camera, a micro injector, and a UV LED source. And so what we did was with the LED off, you have a low pup retrieval during the 12-hour co-housing period. With the LED on, 75% animal retrieving. And so these results look very similar to that of Bianca Jones-Marlin's opto results. So we can confidently say oxytocin is driving this process in the auditory cortex. Now I wanted to move towards activating non-neuronal tissue, because the bulk of oxytocin that is produced in the brain is released into the bloodstream. And so, for example, in the uterus, through childbirth and contractions, and in mammary gland during lactation. And so, for example, the question here is, there's two different cell types, majority cell types that drive the lactation process. So for example, basal cells and duct cells, and they coordinate together to release milk into the baby's mouth upon suckling. And so what actually happens is you have a morphological change that is oxytocin dependent, change that this torsion and pushes out the milk through the nipple. And so these two different cell types are doing the work, but how is relatively unknown. So here I'm just showing you that mammary tissue expresses tons of oxytocin receptor. You can see that here in red, and if you have oxytocin receptor knockout, you have no receptor. And so this is just a wash-on of oxytocin in GCAMP expressing mammary tissue ex vivo. And what you can see here is that you have tons of calcium signaling, and you have some type of synchrony that's going on. And all of the cells moved, and they changed their morphology. Now, the question here is, does oxytocin need to interact with every single one of these clusters to drive this process, or is it enough to start a cascade if you activate only one? Pharmacologically, this is very hard to do. And so what if we use our system to just uncage locally? And we could do that, and uncaging the oxytocin only in this cluster did not propagate the synchrony across the entire system. And so now what we would want to ask down the line is, what if we only activated a single cell within that cluster? Is that enough to drive the process? We're not there yet, but we're getting closer. And so this is just the control. If you just flash with light and saline or a scrambled peptide, nothing happens. And so just a translational aspect of this is that there is a hypothesis that when oxytocin is given to induce labor, it may reduce the oxytocin receptor in mammary tissue, and thus affect a subset of women in their ability to push out milk in the early hours post-birth. And so what we show here is that this is saline injected mice, and then after 5 hours, we look at compare that with oxytocin injected mice. As you can see here, there's a major reduction in oxytocin receptor on the membrane, and this is probably why you have this reduction in lactation. So we wanted to see if we can locally uncage in the uterus to induce contractions without affecting the mammary tissue. But we're not fully there yet, but this is just an experiment to demonstrate that we can do this uncaging of OT in the uterus, and we have this contraction in a dish ex vivo, and we've started to do this under in vivo conditions under ultrasound. All right, so for my future directions, I'm very interested in deciphering the crosstalk of oxytocin and vasopressin systems. Oxytocin and vasopressin are both related peptides, both 9 amino acids, and that are implicated in various social behaviors and physiological processes. And the interesting thing is that, for example, in several brain regions, you will find the overlap between the two. So for example, here in the CA2, CA1 regions, you have an overlap between oxytocin and vasopressin receptors, and there are fibers for both input into this area. However, how do you manipulate both of them at the same time? Turn one on and turn one off so that you can follow the behavior over time. So here I wanted to use a two-caging system. So where you have an oxytocin receptor specific agonist or a vasopressin receptor antagonist, and you can uncage them with a single wavelength of light, turning on one population and turning off another and following behavior. So for my future lab, the goal is to decipher the molecular logic of neuropeptide signaling and control of behavior and physiology. And I want to start with the oxytocin and related vasopressin system, look at what cell types they act on, and then how these circuits converge and diverge to, for example, control behavior and physiology. And also how do they control different peripheral organs and what is the feedback between the two? And then the potential, for example, using them to treat neuropsychiatric disorders. And if we can get that far, then we can use chemistry to modify oxytocin and vasopressin to increase its bioavailability, longer lasting, and specific targeting. With that, I would like to thank everybody who's helped with the work and the funding mechanisms, and thank you all for tuning in. I'm happy to take any questions.
Thank you, Ismail, for this beautiful talk. Can you please stop sharing this screen? Yes,
Yeah.
thank you. Sunny.
Thanks, Dr. Ahmed, for your presentation. Questions from our audience. Start with this question. As you pointed out, Dr. Ahmed, vasopressin and oxytocin are known to be promiscuous with each other's receptors. How do you control for off-target effects of the caged oxytocin on vasopressin receptors?
Yeah, so the experiments that were done here were primarily in areas that do not have any vasopressin receptors. However, we have a next generation TGOT, caged TGOT, which will only or mostly at over 1,000-fold bind oxytocin receptor over vasopressin receptors. So those are some of the things that we have in the pipeline I didn't show here. So we have agonists and antagonists that are specific for each receptor, and we can use them in tandem, either same color or two-color uncaging. There are other strategies as well, but this is just one of the few tools that we're developing to control the oxytocin and vasopressin system or track them.
Thank you for clarifying. Another question is, when activating a single cluster mammary tissue, do you see signal transduction waves similar to what you see when all the clusters in the mammary tissue are activated?
Can you repeat that first part again?
Yeah, sorry about that. When you activate a single cluster mammary tissue, do you see signal transduction waves similar to what you see when all the clusters in the mammary tissue are activated?
No, it's significantly decreased. And we're trying to figure that out. What does that look like? And so a next generation of this tool is looking at 2-photon uncaging using a different caging group so that we can really have very precise control. And maybe, for example, we can uncage at multiple spots at the same time and then see how that drives the process. Yeah.
Fantastic. Thank you for sharing. Are there specific biochemical pathways that are activated by oxytocin when binding to your cell type of interest?
So, for example, right now what we're trying to figure out is, let's say, for example, looking at the pup retrieval behavior, it seems to act on two different inhibitory neurons, SST and PV neurons. And what we're trying to do is figure out which one is oxytocin driving to enhance that maternal behavior. For that, I'm developing a different tool where it's basically derived off of Mike Tadros's DART system, which is basically a tethering system of the drug so that, for example, I can express a flag tag in, like, let's say, for example, the SST Cre neurons, and then that would just like chronically drug or deliver oxytocin to that. and then we can do that puck retrieval assay. So you can't always, like, for example, with this, cell type specificity is very hard because, for example, you would need very fine light in vivo. We can do this pretty well in ex vivo, but to do this in vivo, you would need some type of tethering system.
Thank you for sharing. We have time for one more question. With regards to oxytocin acting directly on the memory tissue, are you aware if there are peripheral neurons that are being activated as well to trigger potentially memory remodeling?
So, I've talked to a lot of some of the top researchers in the memory field, and they know very little about the peripheral neurons that are innervating there. I feel like that's there are definitely several sensory neuron types there, but what they do to coordinate, lactation or whatnot is relatively unknown. And I think that that's one of the questions that I would like to go after is, what is that neuronal, connection? But what we do know is that the oxytocin is not delivered, through a neuronal system to the oxytocin, to the memory tissue. It's released in bulk through the blood, and this is why using a cage oxytocin system gives us that, spatial temporal control in a situation where, the molecule is diffusing in the blood.
Fantastic. Thank you, Dr. Ahmed.
You're welcome.
All right. So our next speaker is Dr. Jonathan Borland. Jonathan received his doctoral degree in neuroscience from Georgia State University and is currently a postdoctoral fellow at University of Minnesota, where he studies the neural mechanism behind the rewarding properties of social interactions. His long-term interest is to understand how the brain encodes the rewarding properties of social interactions, focusing on sex differences, aggressive, and sexual interactions. Jonathan, please go ahead.
Yes, thank you. So yeah. So I think everything's set up and everyone can hear me. So I'll begin. Hello everyone. Thank you for this award. And thank you for the opportunity to present today and to everyone in attendance. I'm a research scientist. And one of the things that interests me as a researcher is what are the neural mechanisms, specifically what are the chemical and molecular properties mediating the rewarding effects of social interactions. Now, social interactions are critical for survival and reproductive success. For example, this bottom image here is of Japanese macaques huddling in winter to improve thermoregulation. Moreover, in humans, mental disorders are estimated to cost the U.S. approximately $300 billion in lost productivity. And social interactions are a powerful factor for improving mental health. Now, to carry out my research, I choose to use Syrian hamsters. Well, why Syrian hamsters? Well, because like many species, including primates, but unlike most rats and mice, both male and female Syrian hamsters display robust dominant subordinate relationships. Dominant subordinate relationships are foundational to the formation of social hierarchies. Thus, they're critical for resource allocation, resilience to social stress, and thus good physical and mental health. For example, I'm about to show you a video of a female hamster defending its home territory from intruding male. Here right now, what you can see is the male, which can be identified by the shaving of fur on its lower back. And directly behind the male is the female. Now, the female is going to target and attack the lower left and right flanks of the male, as this is where both male and female Syrian hamsters have flank glands, pheromones. Thus, due to reliability and robustness of their social behavior displays, and encoding in reward, coding of social reward in both males and females, they serve as the ideal model for studies of sex differences and the rewarding properties of social interactions. Now, I'm specifically interested in the rewarding properties of social interactions because reward has powerful effects on behavior. If something is rewarding, you're more likely to do it again. Now, while a lot is known about the rewarding properties underlying various behaviors, such as eating, drinking, drug use, and exercise. Comparatively, much less is known about the neural mechanisms or rewarding properties of social interactions. Now, surprisingly little is known about the neural mechanisms of social reward because deficits in social reward are associated with many psychiatric disorders, such as social anxiety disorder, depression, and autism spectrum disorder. Finally, it's important to note that not just the quantity, but also the quality of social interactions is important, as the formation of social hierarchies determines the allocation of resources and reproductive success. Thus, the reward properties of social interactions are critical for the expression of adaptive social behaviors and the development and maintenance of beneficial social relationships. Now, the most common procedure for measuring social reward in laboratory context is the Skinner-Place preference test. The concept behind the CVP test is that a particular stimulus is rewarding, in this case, a social interaction. Then subjects spend more time in the environment associated with the rewarding stimulus. However, conditional place preference is just one approach for investigations of reward. Thus, while as a graduate student, I designed and validated a novel operant social preference test that assesses the motivating properties of social interactions. In brief, the arena consists of a main chamber and two smaller adjacent chambers, which can be accessed by pushing through a vertical swing door that can only open one way. Visual and olfactory cues guide the subject to the stimulus hamster, and there's also a bucket on the back of the door that can be used to hold weights. Here's a video of a subject's first time inside this apparatus. For time's sake, this video has been sped up to 8x. Now, while the subject here is allowed to move throughout the apparatus, the smaller same-sex stimulus hamster is confined to the left or the right smaller chamber. Now, one of the things I want you to note is all the exploratory behavior in the beginning. The subject has no previous experience in this arena. The vertical swing doors, or lies on the other side of the doors. Now, 20 seconds after entering into either small chamber, the subject is returned to the main chamber. Importantly, unlike in the CV test, the subject has the choice whether or not to engage with a social stimulus. Another thing I want you to notice is that the latency to re-enter the social chamber drastically decreases as this test session progresses, demonstrating that the same-sex social stimuli serves as a great motivator and reinforcer for performing this task. Social preference can be quantified as the number of entries into the social chamber minus the number of entries into the empty chamber. Furthermore, by adding weights to the door, I can increase the behavioral cost requirement to access the social stimulus. I measure the time subjects spend attempting to open the door as an alternative measure of social motivation. Next I want to ask, so what circuit regulates social behaviors? Well, as discussed earlier by Dr. Ahmed, the most well-known system implicating social behaviors is the oxytocin system. Oxytocin is a 9-amino acid peptide that is synthesized in the hypothalamus and is released during childbirth to increase uterine contractions and involve and lactation in various social behaviors. For example, it also increases eye contact in a patient with schizophrenia and is responsible for variables that form lifelong partnerships with their mate. Thus, oxytocin has taken on the dogma of being the lorphone. Thus, for my dissertation at Georgia State University, I investigated the effects of oxytocin system on social reward. Now, the principal circuit for reward encoding is the mesonomic dopamine system, which primarily consists of dopamine neurons in the visual mental area that sends projections to the nucleus accumbens. And interestingly, as been shown, the oxytocin expressing neurons in the paraventricular nucleus of the hypothalamus, the PVM, sends projections to the VTA. Now, a few years ago, Dr. Ziman or Eric Song performed a study which showed that the oxytocin system in the VTA is necessary for the rewarding effects of social interactions in males. The y-axis here is the time spent in the chamber associated with social interaction, and OTA is an oxytocin receptor antagonist. However, almost nothing was known at the time about how the oxytocin system regulates social reward in females, or if there are real sex differences in social reward. Thus, for my dissertation, I chose to investigate this gap in the literature. I first investigated if oxytocin receptors in the VTA are necessary for social interaction to be rewarding in males and females. Selective oxytocin receptor antagonist was microinjected in the VTA, part of 3 conditioning sessions of the CV test. Focusing on just the black bars first, with just saline treatment. Females are displaying greater social reward compared to males. And interestingly, injection of an oxytocin receptor antagonist in the VTA significantly reduced the rewarding properties of social interactions in both males and females. I then investigated the effects of injecting oxytocin in the VTA and social reward in males and females. Now, whereas in males, injection of oxytocin in the VTA increased the warm properties of social interactions compared to controls. In females, the same dose of oxytocin decreased the warm properties of the interactions. Now, hypothesizing this effect is an inverted relationship between social reward and the duration or intensity of social stimulation, with females being shifted left the curve relative to males. Finally, translational work in humans from Dr. James Rilling's lab at Emory University has shown that during a Churchill competition test, intranasal oxytocin increases fMRI blood oxygen level dependent activity in the caudate putamen in males compared to placebo. The caudate putamen is adjacent to nux accumbens. But in females, intranasal oxytocin decreases blood activity in the caudate putamen and in the VTA during social interaction compared to controls. It is also important to note that at baseline with just placebo treatment, females are displaying greater intensity of blood activity in the caudate putamen compared to males. Thus, there may be an inverted relationship between social reward value and the duration or intensity of social interaction. I believe this work has the potential to improve our understanding of fundamental differences in how males and females encode the warring properties of social interactions, and to provide a better understanding of sex differences and susceptibility to neurological disorders and the clinical treatment. For example, if females are more sensitive, their curve is shifted to the left, and will explain why females are more prone to disorders associated with social stress, such as social anxiety and PTSD. And if males are less sensitive, if their curve is shifted to the right, and may help explain why males are more prone to disorders associated with social anhedonia like autism. Now, following my graduate research, I'm now a postdoctoral researcher at the University of Minnesota. I'm now studying the molecular changes occurring in the nux accumbens from social interactions, particularly focusing on plasticity and synaptic communication. Now, the central nervous system controls behavior via the electrical connection and communication between neurons. Those changes in electrical activity between neurons are going to underlie changes in behavior. And the part of the neuron that has been sensitive to these changes is the synapse. Those changes in synaptic structure in neurons in the nux accumbens should underlie the encoding of reward. Furthermore, glutamate receptors in particular are characterized by significant fluctuations in expression relative to changes in the activity or physical structure of neurons. Those changes in the expression of glutamate receptors in nux accumbens should underlie reward. And to investigate this, female Syrian hamsters had 5 consecutive days of social experience. And I investigated the protein expression of different subunits of glutamate receptors. Now, in addition to there being an increase in the protein expression of post-synaptic density 95 compared to controls that had no social experience, I also found there was an increase in the protein expression of the dimer form of mGluR5 in nux accumbens, a metabotropic glutamate receptor. Aggressive experience also results in an increase in the average subunit GluA1. And so these changes in glutamate receptor expression likely regulate an increase in excitosynaptic transmission, potentially encoding reward. However, I next want to investigate what second messenger signaling molecules inside of the neuron and the nux accumbens associated with the glutamate receptor activation may mediate these changes. I also found that there's an increase in the phosphorylation of ERK1/2 in the nux accumbens following 2 days, but not 5 days of regression compared to controls. Now, interestingly, ERK1/2 or extracellular signal-related kinase is a molecule that regulates the activity of transcription factors such as CREB and CFOS, and is thus responsible for the growth and proliferation of neurons. Thus, these changes in glutamate receptor expression and second messenger signaling likely regulate changes in genetic structure and strongly support an increase in cytosynaptic transmission, potentially encoding reward. So to review, social reward takes the shape of an inverted function mediated by the dose, duration, or intensity of social stimulation and oxytocin activation. And females curve may be shifted to the left relative to males. Thus, both sex and dose should be carefully considered if drugs targeting the OD system are developed to treat social reward-associated disorders. Aggressive experience results in an increase in PSC95, mGluR5, GluA1, and the phosphorylation of ERK1/2 in the nux accumbens. These studies advance our understanding the molecular mechanisms that may regulate the reward properties of social interactions. Furthermore, these studies have provided a foundation for my independent research career. My immediate future goals are to investigate the effects of social experience on synaptic plasticity in nux accumbens in male and female Syrian hamsters, investigating sex differences in sensitivity and mitral sex differences in oxytocin modulation of synaptic plasticity. Currently, I'm training in fibrophotometry to investigate the effects of social experience on the activity of calcium in males and females. I hypothesized that social experience will result in potentiation of calcium activity in nux accumbens, and this potentiation will be greater in females compared to males. Also utilizing optogenetics to investigate the role of glutamate projections from the prefrontal cortex to the nucleus accumbens on calcium activity and other warrant properties of social interactions in males and females. This is an image of the expression of GCAMP in green in the nucleus accumbens and the opsin crimson bar in red in prefrontal cortex projection fibers in Syrian hamsters. And this is an example trace of calcium activity in the nucleus accumbens and the average calcium response to the initiation of social interaction in Syrian hamsters. Furthermore, I also plan to utilize Cre-dependent viruses to dissociate the role of direct pathway and indirect pathway medium-spotting neurons in social reward. Direct and indirect pathway medium-spotting neurons are the two main cell types that are hypothesized to have opposite effects on reward. Finally, I'm also gaining experience in ex vivo selective physiology to investigate specific mechanisms of synaptic transmission in neurons in males and females. Thus, for the start of my independent research lab, I plan to then apply these approaches to investigate the role of oxytocin projections from the PVN to the nucleus accumbens on social reward. These are images of the expression of GCAMP and oxytocin neurons, which are in red, and the PVN in Syrian hamster. The GCAMP expression is selective for oxytocin neurons, and this selective expression is being driven from a viral vector that contains the promoter sequence for oxytocin. I'll examine the activity of calcium in oxytocin neurons in the PVN and utilize viral opsins to investigate the role of oxytocin from the PVN to nucleus accumbens circuit on social reward. Collectively, from these research experiences, there are three main pillars of research I wish to pursue as an independent investigator. I first want to investigate the circuit, synaptic, and intercellular mechanisms mediating sex differences in their warrant properties of social interactions. I also want to investigate the mechanisms mediating the valence of social interactions, mechanisms governing an increase in reward, a decrease in reward, or aversion. And investigate differences in the mechanisms mediating the warrant properties of social and the warrant properties of sexual interactions compared to aggressive interactions. Previous research supports differences in the normal mechanisms in the nucleus accumbens for different types of social behaviors. I'd like to thank my graduate mentors, Dr. Elliot Aubers and Dr. Carl Franz, and all the members of the lab. And also like to thank all my postdoctoral advisors, Drs. Robert Mizell, Parmal Singh, Patrick Rothwell, and the members of the lab. Thank you. Yeah. Questions? I guess not. Stop sharing.
Dr. Borland, thank you for your presentation. That was really insightful. I had a few questions to start. Please clarify with hamsters. With your studies, were they single housed or were they group housed?
They are single housed. The subjects are single housed, and the stemless hamsters are group housed to reduce the levels of aggression.
Okay, thank you. And in the natural environment, are hamsters generally are they relatively independent animals, or are they in herds or packs, whatever the phrase may be?
So they come from the fields of the Aleppo fields in Syria, and their natural habitat. They live in isolation, but they still seek each other out to find mates and still do engage in social interactions. Yes.
Thank you.
Thank you.
I'll ask another question while we wait for more questions from our audience. With regards to one factor I was curious with regards to your studies was level of activity. For example, metabolic metabolism, and if there's increased activity, how that may influence oxytocin production, and then the observations you're seeing. So I was curious to hear your thoughts on just general activity levels and how that may have an effect on your research hypotheses.
Like in like the whole system metabolic activity.
For example, if you were to introduce a wheel with in the cage of the hamster, and the hamster were to run on that wheel, and afterwards, how would that may have a potential effect based off their increased level of activity?
Yeah, very so in terms of reward processing, it's interesting that, a lot of different behaviors sort of interact at the nucleus accumbens. And so, so whether, so both exercise, drugs of abuse, and social interactions, they can all produce rewarding and reinforcing properties. And so I would predict, so similar to how social interactions can reduce or interact with the rewarding properties of drugs of abuse, exercise may interact with the rewarding properties of social interactions and vice versa. There may be a limit in terms of the plasticity in the nucleus accumbens in that regard.
Thank you for clarifying, Dr. Borland. We have a question from our audience. Could you comment on the genetic tractability of Syrian hamsters? Is anyone, are there transgenic CRISPR lines?
There's at least one. There's at least there is one's transgenic line of a vasopressin receptor knockout Syrian hamsters. And in terms of, and that's been used for investigations of social behavior. I also know that Syrian hamsters are used a lot for a lot of cancer research and also research in doing with metabolic activity. And so on the more biology aspect in that field, I'm not quite sure in terms of what's available, but there. But yeah, that's currently sort of in the development.
Thank you. I'll ask one more question. So you mentioned having interest with neurocognitive conditions in the context of your research studies. Are there differences, and this is a very general question, are there differences in oxytocin concentration that may affect the observations that you are seeing, and then are those potentially also involved with other differences in sex as well, in terms of oxytocin concentration that may influence your experience as well?
Yeah, that's great. Yeah, great to hear. A really interesting question. So yeah, there could be differences, in sort of baseline endogenous levels of oxytocin and its role on social behavior. What I think is most interesting is that, sort of the companion peptide of oxytocin is vasopressin. If anything, the research has shown that in terms of endogenous expression of the neuropeptide vasopressin and differences in levels of vasopressin receptor expression, that vasopressin is actually more sexually dimorphic than oxytocin. And then also oxytocin may potentially be more involved in sort of the regulation of social reward, while vasopressin may be more involved in sort of the expression of social behaviors. But yeah, that's definitely something that needs to be considered and investigated, is differences in reward processing. And for different species, if there's sex differences in different brain regions and endogenous levels of both oxytocin and oxytocin receptor. So there are some, but if anything, the effects are more dramatic with vasopressin.
Thanks, Dr. Borland, for clarifying and for your presentation again.
Yeah, thank you.
So our next speaker is Dr. Annie Raiz. Annie is a neuropsychology postdoctoral fellow in the Department of Radiation Medicine and Applied Sciences at UCSD. She is interested in examining cognitive diagnostic frameworks in epilepsy and brain tumors, and understanding and reducing health disparities across neurological disorders. She aims to develop a health disparities research program and a neuropsychology clinic for providing culturally informed neuropsychological services to bilingual and Spanish-speaking patients.
Thank you for the introduction, and thank you for this opportunity to present here regarding my research and current research and future plans. As I mentioned, I'm a postdoctoral fellow in the Department of Radiation Medicine and Applied Science at UCSD. I have a PhD in clinical neuropsychology and have been trained in advanced statistics, neuroimaging, health disparities research. I'm currently funded by NIH Diversity Supplement and by the Burroughs Wellcome Fund. And I have over 10 years of experience working with clinical populations, including epilepsy, brain tumors, and dementia. And my work has largely focused on using neuropsychological and neuroimaging tools to investigate the cognitive and psychiatric comorbidities of neurological disorders. And today I'll be discussing some of my current research and the research program I plan to pursue as a faculty. So epilepsy is the fourth most common neurological condition, and it is associated with significant economic burden. The average cost ranges from $8,000 to $11,000 per person, and the cost rises to over $25 billion a year with uncontrolled seizures and comorbidities. Most of the cost of epilepsy is associated with the treatment of these comorbidities and the impact of the condition on social outcomes, including education, occupation, and income. This burden greatly impacts families with low socioeconomic status and those living in rural areas. Importantly, epilepsy is associated with significant quality of life burden that impacts the individual living with epilepsy and their families. Given this social and economic burden, I have spent the last 10 years investigating the impact of epilepsy and epilepsy treatments on cognitive and psychosocial outcomes. I have led or co-led several studies exploring different approaches to characterizing cognitive impairment, including phenotyping, identifying unique clinical profiles that can help identify patients at most risk for cognitive decline, and understanding the impact of non-epilepsy factors on cognition, including bilingualism and vascular risk factors. And then more recently, I have been developing a research program focused on identifying social determinants of health in epilepsy, including socioeconomic disadvantage, and examining their impact on cognitive and psychosocial outcomes. I'm now expanding this work by focusing on addressing gaps in access to care by developing community-informed interventions. There's significant disparities in epilepsy, including higher rates of epilepsy among non-white individuals and those from disadvantaged backgrounds. This is access to specialized care, including diagnostic tools, more emergency room visits, and generalist providers. And then there are disparities in the treatment, including lower rates of surgery among non-Hispanic, Black, and Hispanic Latinx individuals. And then lastly, patients from underserved populations have higher rates of comorbidities that are often not treated. Several factors have been identified to be associated with these disparities, including socioeconomic status or SES, health insurance coverage, education, occupation, and income, health literacy, and then language barriers. Despite this, there are few studies examining the impact of these disparities on brain, cognitive, and overall health outcomes. And then among individuals with epilepsy, older adults require comprehensive disease management, given that they present with multiple comorbid conditions. These include risk factors for accelerated cognitive and brain aging, including vascular and metabolic risk factors, mood disorders, ultra lifestyles, and poor quality of life. And there's also shared pathology between patients with epilepsy and Alzheimer's disease that include AD-related pathologies such as amyloid plaques and tau tangles. These combination of factors place individuals with epilepsy at increased risk for developing dementia, including vascular and Alzheimer's disease. And despite this, seizures are often an exclusion criteria in clinical trials of Alzheimer's disease and related dementia. And then lastly, despite these elevated risk factors and accelerated cognitive aging, only recently has there been an increased interest in examining the cognitive profiles of older adults with epilepsy. Given the importance of early detection of cognitive decline, my work has focused on applying novel cognitive diagnostic criteria to older adults with epilepsy and identifying factors associated with cognitive dysfunction. In a study published in Epilepsia, I applied a mild cognitive impairment or MCI criteria to a group of older adults with epilepsy and found that approximately 60% of the sample met this criteria with profiles that were similar but not identical to individuals with amnesic MCI, the preclinical phase of Alzheimer's disease. Specifically, patients with epilepsy had greater impairments in language, while those with MCI had greater impairments in memory. And then in a more recent study published in Frontiers of Neurology, I applied my expertise in cognitive phenotyping to a community-based cohort of older adults with late onset epilepsy and found that about 63% of the sample demonstrated an impaired cognitive phenotype, with approximately half of the participants declining at a follow-up visit. Importantly, higher education and occupational attainment was associated with a minimal or non-impaired profile at baseline and with a lower likelihood of declining over time. This highlights the impact of social determinants of health, such as education and occupation, on cognitive outcomes. I'm also investigating the role of neighborhood socioeconomic disadvantage in cognitive outcomes by examining the relationship between the Area Deprivation Index, or ADI, which is a multifaceted proxy measure for assessing socioeconomic disadvantage at the neighborhood level, and cognitive performance in older adults. We found that higher ADI values, which indicates greater disadvantage, was associated with poor scores across measures of learning, memory and processing speed, and executive function, and that patients residing in the most disadvantaged neighborhoods were younger, had few years of education, had lower personal and family incomes, had lower father education, and were taking more anti-seizure medication. Importantly, neighborhood disadvantage is a potential modifiable social determinant of health that could inform epilepsy-related policy aimed at identifying the most at-risk neighborhoods and then developing system-level interventions focused on increasing access to epilepsy care, education, as well as resources. And then these independent patient cohorts had an elevated number of modifiable dementia risk factors, including hypertension, obesity, diabetes, and hyperlipidemia, with a history of smoking and hypertension the most common. Despite the alarming risk for dementia and the emergent evidence demonstrating the increased cerebrovascular disease burden in older adults with epilepsy, there's virtually no literature identifying ways that patients can reduce the risk for cognitive decline and dementia, particularly by targeting modifiable dementia risk factors. So to address this immense gap in disparities in care research, I am developing a health equity research program focused on older adults with chronic medical conditions, including epilepsy and cancer, where I plan to integrate the newly published Social Determinants of Health framework from the National Institute of Neurological Disorder and Stroke, or NINDS. Specifically, my lab will focus on investigating the impact of structural determinants, including economic, public, and social policies. I will also continue to look into social status determinants that include education, occupation, and income. I am particularly interested in investigating the role of access to care in health inequity, particularly among older adults. At the individual level, health behaviors and knowledge, as well as psychosocial factors, offer the perfect opportunity to develop interventions that are aimed at providing patients and their family with tools and knowledge to better manage their condition and improve their well-being. And then lastly, I will continue to use my expertise in neuropsychology and neuroimaging to examine the interplay between the social determinants of health and biological determinants of health. So my lab will be named Brain Core, or Community Outreach Research and Engagement to Promote Brain Health Equity. The initial two aims will be to examine the impact of social determinants of health on cognitive and health outcomes in individuals with neurological conditions, including epilepsy, and then to develop community-informed interventions addressing disparities in access to care. The health equity lens of my lab will focus on social, structural determinants, health policy, and social needs and risk. I will use community-engaged research methods, including dissemination and implementation science, and community-informed research design. The research will be translational, which means that the end goal will be to develop community-informed interventions to address these unmet needs, with some target goals including improvement of early detection of cognitive decline, improvement of mood and quality of life, and then addressing social needs through resources and support. And in order to obtain these skills and knowledge in community-engaged research methods and intervention development to run an independent lab, I recently submitted a K-23 to NINDS, where I'm proposing to develop the Health Aging in People with Epilepsy program, or HAPPY, which is a community-informed educational program for older adults with epilepsy. In this program, I will be targeting epilepsy management, including medication management, knowledge of surgical options, treatment of mood disorders. I will also integrate dementia management, including increasing dementia knowledge with the goal of improving early detection, And then lastly, I will integrate chronic care management models to address modifiable vascular risk factors, including hypertension, hyperlipidemia, treating diabetes, and so forth. The reason why this model is needed of an integration between epilepsy management and dementia management is that patients with epilepsy have very unique cognitive needs. A lot of these patients have cognitive impairment due to their seizures, but they're also aging, which is putting them at risk of developing something like Alzheimer's disease. So special attention is needed for this population to make sure that we are developing programs, educational programs that are targeting these needs. Importantly, stakeholders, including patients and providers, will be involved in the program development. The program will be offered both virtually and in person, addressing the transportation needs of patients with epilepsy. And then lastly, I plan to linguistically and culturally adapt the program in Spanish for Hispanic Latinx and test the preliminary feasibility, acceptability, and impact of the program. Then to ensure successful adaptation and implementation of the intervention, I will use the Menary Vanderbilt Community Engagement Studio to obtain feedback from stakeholders on topics related to aging and epilepsy, unmet needs, and availability of local and hospital resources. This information will be developed, it will be integrated to the development of the final program. I will then pilot the program, as I mentioned, to examine its feasibility and acceptability. And this will serve as preliminary data for an R01. If the K-23 is awarded, I will be working on this project in the summer of 2024. And then other research projects I plan to pursue once I become faculty include a longitudinal study where I plan to examine the impact of social determinants of health on cognitive and brain trajectories in patients with epilepsy. These will include structural system level factors, early life stressors, and individual patient factors. I plan on submitting an R01 to collect cognitive and neuroimaging data in ethno-racially diverse and socially economic disadvantaged cohorts. I also plan to develop and submit a multi-center R01 investigating the cognitive and imaging profiles of Spanish-speaking Latinx patients with epilepsy. This community is underrepresented in epilepsy research, and some of my work has presented as the first studies in this clinical population. I will use community-engaged research methods for the recruitment and retention of participants, but importantly, participants and their family will be provided with patient education to improve management of their condition. And then lastly, I am interested in identifying barriers to recruitment and retention of historically and excluded groups in clinical research by working closely with community stakeholders to identify barriers as well as needs and develop community outreach programs that includes education on brain health. I will develop culturally informed and ethical recommendations on recruitment and retention of historically excluded participants in communities in biomedical research. And my lab will serve as a consultation team, not just for the institution that I am part of, but other researchers who are getting funding from the NIH. For this study, I will be planning to submit an R21. So I want to just quickly thank all of my mentors and collaborators, as well as the Lab of Cognitive Imaging LOCI that is led by Dr. McDonald at UCSD, and my funding sources, which includes NINDS, the National Academy of Neuropsychology, and as I mentioned earlier, the Burroughs Wellcome Fund. Thank you.
Thank you, Annie. Do we have any question?
Yes, Dr. Reyes, thank you so much for your presentation. One question from my audience is if for your findings, insights on epilepsy-related disparities, if they're relevant to a wide range of different neurological conditions, possibly.
Yes, so my screen is frozen. Okay, I'm back. Okay, there we go. ADI, the Area Deprivation Index, which is part of the Wisconsin School of Public Health, has been actually used pretty widely in other studies. One of the major studies was in COVID-19, and what did they find is that individuals who lived in neighborhoods that had higher ADI or the most disadvantaged did have higher rates of COVID and particularly more COVID complications. Also, those living, there's been studies in Alzheimer's disease and related dementia as well, showing that greater neighborhood disadvantage is also associated with higher rates of dementia, and specifically Alzheimer's disease.
Thank you for clarifying. The follow-up question being, you highlighted that it's a multimodal, multifactorial context that you're addressing. How could you elaborate on how you'll be considering that with regards to the different implementation therapeutic interventions that you have planned? You mentioned exercise, you mentioned diet, you mentioned various potential therapies. Where do you plan to start?
Yeah, so it's going to be an educational program. So meaning that what we're planning to do is first survey literature in terms of all of the epilepsy, individual epilepsy, as well as dementia educational programs, interventions that are out there. And then really kind of get these topics and survey patients and providers to kind of see what are the most interesting topics that the program development would include this information. So it's going to be specifically educational focus, meaning that we're going to provide backgrounds in terms of how to manage your condition, the importance of exercise and diet into improving brain health, or what to do, what are some of the early signs of cognitive decline, and if so, if you're experiencing these, what is it to do next? Talk to your provider, talk to your epileptologist. It's going to be a pilot study with the goal of
Dr. Reyes? Our speaker is potentially having some connection issues. Okay. Well, please welcome to follow with Dr. Reyes with any additional questions. But we thank you for your presentation. Dr. Das, would you like to continue?
Yes, thank you. So our final speaker of this session is Dr. Erica Rodriguez. Erica obtained her PhD in neurobiology at Duke University. She joined Columbia University as a Helen Hay Whitney and NIH T32 postdoctoral fellow and is currently a Mosaic K99 ROO scholar. She is interested in understanding how neural processing of social sensory cues impacts emotional behavior. Her future goal is to uncover the contributions of neural circuits and neuromodulators to normal and impaired social processing of autism spectrum disorders and schizophrenia. Erica. Erica, can you please share your screen?
Sorry, my dog is barking.
That's all right.
And my phone
Share your
is noises. Give me one second. Okay, everything good? Okay, sorry about that. This is what happens when you're presenting at home. Thank you to the organizers and the selection committee for giving me this opportunity to present my work today. As was stated in the introduction, my name is Erica. I'm a neuroscientist working at the Zuckerman Institute at Columbia University with Dr. Daniel Salzman. And today I'm excited to talk about the neural mechanisms underlying social motivation. So day to day, you have to constantly decide how to behave appropriately based on your changing social environment. So for example, your various social interactions with these two individuals may impact your motivation to continue interacting with them. As a researcher, I am fascinated by the ways in which these social interactions shape decision-making processes in both humans and other animals when they are experiencing positive or negative events. More specifically, I'm using mice as an animal model to investigate the role of olfactory cues in guiding social motivation. And with this model, I hope to gain a better understanding of the neural mechanisms underlying social decision-making and to identify potential targets for interventions aimed at improving social functioning in humans. And so social interactions give rise to social constructs. So for example, whether the perceived individual is male or female, friend or foe, dominant or submissive. And these social constructs inform our emotional responses to others and guide our behavioral decisions such as approach or avoidance. To understand how social interactions give rise to these constructs, we must first understand how sensory information is represented about other individuals and how social meaning is assigned to these neural representations. In my line of research, I am interested in understanding how the social construct of hierarchy modulates emotional processing and motivational behavior in mice, and whether this generalizes across male and female groups. And so how social information directs emotions and behavior remains poorly understood as we lack the mechanistic insight into the neural pathways involved in the conversion of sensory representations to social behavior. And so the goal of my project is to define the neural circuits that process social olfactory cues to coordinate and shape that social behavior. Although there's rich literature focused on rodent responses to neutral odors associated with threats, predatory and opposite sex odors, there has been little focus on odors with socially significant meaning within same species or conspecific social groups, in particular with regards to social hierarchy. There is emergent evidence that the anterior cingulate cortex and the basolateral amygdala may play a role in encoding information about conspecifics. So both the ACC and BLA are part of the emotional processing system of the brain and are involved in the acquisition and expression of directly learned emotional responses to prior neutral sensory stimuli, as well as innate responses to emotionally significant stimuli. And so building from this knowledge, recent literature has shown that social odor and rank are both represented in the ACC and BLA, and that the ACC to BLA pathway is involved in observational learning, which is the ability to learn about threat or reward predicting events by watching other individuals directly subjected to these appetitive or aversive events. Additionally, work in our lab has shown that social and motivational information is segregated in the ACC, but converges in the amygdala. However, defining the precise neural circuit mechanisms by which these distinct representations in the ACC might interact with overlapping neural representations of social and non-social value in the amygdala is extremely challenging in the monkey experimental prep. Therefore, we've been using the mouse to determine how a representation of socially ranked conspecific odor in the ACC modulates BLA neuroactivity and emotional processing. I first focused on characterizing the behavioral responses to dominant and submissive conspecific odors and compared these responses based on the social status of experimental mice and social stimuli. Before assessing the behavioral responses to conspecific odors, I first have to assess the rank of the mice. So I'm using the tube test, which is a commonly used social hierarchy assay. Loser is the mouse that leaves the tube first, as you can see in this video. I run the test until I have 4 days of consistent ranking, in which then I determine that that group is having a stable hierarchy. And we observed that both males and female groups form stable hierarchies at a similar rate. After ranking the mice, I collect the urine from the highest and lowest ranking mouse, which will be used as well-controlled social odor stimuli. I then measured behavioral responses of the 2 middle ranking mice in this infrared beam break assay that I developed. During the infrared beam break assay, mice undergo a 1-day session broken down into multiple trials. In addition to being presented with urine from either the submissive or dominant mouse, mice are also presented with their own urine as a social neutral odor and mineral oil as a non-social neutral odor. The cotton swab is placed in a holder. It also contains an infrared beam, which when broken would indicate that the mouse is sniffing the cotton swab. We were able to calculate both the frequency and duration of beam breaks or nose pokes, as demonstrated in these average frequency plot traces. When looking at the frequency of nose pokes across time, we saw the greatest differences in the first 2 minutes on each trial, so we decided to focus our analysis in that time window. We saw that male mice investigated dominant odors more frequently and for longer durations than submissive odors, as shown in both these raw data sets and after normalizing the data within each individual mouse to account for differences or bias effects in baseline movement. In contrast, female mice investigated submissive odors more frequently and for slightly longer durations than submissive odors. And in another set of experiments not shown here, we saw that male mice also investigated unfamiliar dominant male odors more than unfamiliar submissive male odors, whereas there was no difference seen in approach behavior in female mice. And so altogether, these findings may suggest that there may be both a learned and innate component to male motivational behavior towards socially ranked odors, whereas there may only be a learned component to female motivational behavior. However, the limitation of measuring with the infrared beam is that it only captures the behavior within the holder, and it fails to capture the animal's behavior beyond the holder. So in conjunction with using infrared beam measurements, I tracked the mouse's position and movements using Deep Lab Cut. Analysis of this data is ongoing, but as an initial first step, I measured the mouse's orientation in the middle of the chamber, shown in red, to determine whether the mouse was oriented towards or away from the odor. And these measurements reveal that male mice spend more time oriented towards dominant odors more than submissive ones, whereas female mice trended towards spending more time oriented towards submissive odors more than dominant ones in the middle of the chamber. And so these findings further strengthen this idea that this olfactory-driven motivational behavior is sex-dependent. As part of our ongoing effort to align and correlate behavior with neural activity, we also plan to identify other behavior repertoires. This approach is critical for identifying neural ensembles that encode aspects of social processing or complex behaviors that are beyond approach or avoidance. And it potentially identifies time epochs in which neural ensemble activity can predict upcoming social behavior. We next wanted to determine whether the anterior cingulate cortex neurons that project to the basolateral amygdala also contribute to this olfactory guided motivated behavior. To determine whether BLA projecting ACC neurons played a role in this motivational behavior, I expressed JAWS, a light silencing opsin into the ACC, and implanted optic fibers above the BLA, as shown in these schematics and images. I then used a modified infrared beam break assay, which incorporated a closed loop optogenetic approach. Using the mouse's positions, an LED light was triggered when the mouse was close to the odor. During sessions with no light, male mice were observed to significantly approach dominant odors, more than submissive ones, as seen in the initial experiments. Similarly, female mice were trending towards approaching submissive odors more than dominant ones. Interestingly, during sessions with photoinhibition, male mice were observed to no longer approach dominant odor, with a trend towards actually approaching submissive odors more, whereas female mice were no longer approaching submissive odor more than dominant ones. And so this data may suggest that the ACC to BLA pathway, which may influence the excitatory and inhibitory balance in the BLA, is needed to appropriately discriminate and behave towards differentially ranked mice. And so these findings have provided the foundation for my ongoing work, in which in a multi-pronged approach, I'm defining the synaptic circuit and population level dynamics in the ACC and BLA that could shape and convert social information into sex variant socially driven motivation. The first set of ongoing experiments explore whether the same neural ensembles that represent stimulus reward associations also represent social rank in the amygdala, and whether social and motivational information is segregated in the ACC. As stated in the beginning of my talk, these questions stem from previous monkey work done in the lab. So it would be interesting to see if this generalizes across species and whether we can gain more insight into using the mouse model. This requires us to measure real-time changes in neuroactivity in the ACC and BLA of freely moving mice during exposure to socially ranked odors using genetically encoded calcium markers and 1-photon imaging. In trying to answer these questions, we would parse out the ensembles encoding for sensory information on social rank and determine whether they correlate with reward value, assess how social sensory information is relayed from the ACC to BLA, and determine how BLA transforms the sensory information assigned with social meaning into motivational behavior. The next set of ongoing work explores how oxytocin and vasopressin, two highly conserved neuropeptides, which Ishmael did a very wonderful job of describing earlier, they play a role in regulating social behavior, how they may contribute to the shaping of social olfactory information. These peptides are known to enhance the signal-to-noise ratio of sensory stimuli, thereby enhancing neural signal transmission and possibly strengthening neural synchrony, which results in conveying the saliency of social stimuli. In trying to answer these questions, we are molecularly and physiologically dissecting the circuitry of BLA projecting oxytocin receptor expressing ACC neurons. So initial circuit mapping experiments have revealed that these neurons, which directly send excitatory input into the BLA, may largely be receiving non-synaptic input from oxytocin and vasopressin neurons. Further studies, of course, are needed to be determined whether and how these neurons contribute to the encoding of the different degrees of saliency to socially ranked odors, which results in the sex-specific motivational behavior that I'm seeing in my current data sets. And lastly, in my future lab, I will combine my molecular expertise gained during my PhD with my ongoing postdoctoral training in systems neurobiology to broaden my investigation into how social sensory cues are processed and shaped at multiple levels of sensory, motor, and emotional regulating systems, and to understand how this becomes aberrant in mouse models of autism and schizophrenia. This will include looking at how olfactory information is assigned social meaning in sensory processing regions, as well as looking at how other neuromodulators may be involved in shaping olfactory information. Additionally, I will broaden my investigation of understanding how other sensory stimuli contribute to socially driven motivation, both individually and collaboratively. I believe that understanding each puzzle piece of the social sensory processing will shed light and guide me in designing future studies focused on investigating how mice make social decisions when perceiving conspecifics. This may reveal whether all or some of the sensory processing is aberrant and to what extent in psychiatric mouse models. And of course, there will be a constant underlying theme in my research program investigating the potential gender differences on social sensory processing and behavior. This is based on the emerging evidence that there are sex differences in patients with autism and schizophrenia. Therefore, it's critical to understand and compare normal and aberrant synaptic mechanisms of social sensory processing and social behavior in both male and female wild-type mouse models and psychiatric models. And altogether, this work that will emerge from my research program will provide fundamental mechanistic insights that have the potential to drive the development of personalized novel treatments for psychiatric disorders. And so I would like to thank the Salzman Lab, especially the students that I was very fortunate enough to work with all these years, as well as my collaborators and people who have just helped me along the way. And this work has been supported by the Helen Hay Whitney Foundation, the T32 Training Grant, as well as now the Mosaic K9 R00 Award. And with that, I'll take any questions.
Thank you, Dr. Rodriguez, for your presentation. Really wonderful. We have time for one question, likely.
It's also the end of the day, so.
There's that too. I'll ask a question. With your research, were these adult mice?
Yeah, so these are at least 8 weeks old, 8 to 10 weeks, not more than 3 months. So I haven't looked at adolescence, so I don't know if like development is a factor or if aging is a factor, but those are things that I'm interested in my future lab. So
Absolutely. We look forward to hearing from you in the future. Thank you for sharing.
thank you.
And with that, this concludes the end of this session on brain and behavior. Thank you all for your to all the fellows for their presentations. It was really wonderful hearing from you all. A big thank you to our attendees for participating and engaging. And with that, we will conclude this session, and we'll have Drs. Sohini Banerjee and Maiko Katakato to continue our conversation with closing remarks.
Thank you.
Good evening, everyone. I'm Sohini Banerjee, and I'm a postdoc at the Baylor College of Medicine.
And I am Maiko Kitaoka. I'm a Hannah Gray postdoctoral fellow at the Whitehead Institute. We are the co-chairs of the 2023 ISFS Program Planning Committee. Thank you all so much for joining us for the first full day of ISFS 2023. We'd like to take a few minutes today to recognize the many contributions that go into this organization and program.
The Intersection Science Fellow Symposium was envisioned as a multi-institutional effort to provide postdocs from historically underrepresented backgrounds with visibility, mentors, sponsors, and professional development to mitigate some of the systemic challenges to diversifying academia. So it is critically important to ISFS that postdocs, our future faculty, have a voice and decision-making power in this organization and event. In its current form, the symposium is designed and led by postdocs as a multi-institutional collaboration with advice from the ISFS leadership team and advisory committee. There are 3 postdoc-driven committees that oversees the different aspects of ISFS.
First, the Outreach Committee aims to reach a broad and diverse set of applicants as well as audience members and celebrates our fellows and associates among hiring institutions and scientific audiences via social media and up-to-date email communications. Many of us have probably gotten many emails from them, actually. They also help to keep the website up to date, to recognize all of the ISFS contributors and share professional development opportunities more broadly.
The second committee is the Program Planning Committee, which we co-chair. This committee is responsible for organizing the 3-day symposium. We coordinate with the fellows and associates into the sessions and identify the relevant topics and speakers for the public and non-public events of the symposium. We also coordinate with institutional representatives, faculty volunteers, and mentors for the networking sessions. Finally, we are actively involved in making the symposium run smoothly by recruiting moderators and coordinating run-throughs.
And lastly, the Professional Development Committee coordinates a year-long program of workshops and activities for the fellows, both pre and post-symposium. They facilitate faculty mentor matching, work with DEI leaders to organize mentor-mentee orientations and develop mentoring guidelines, and also identify topics and speakers throughout the year. You'll hear more from both the Outreach and Professional Development Committee co-chairs on day 2 and 3 of this year's symposium.
As you can appreciate, that planning an enterprise of this breadth and scope involving multiple institutes and individuals in different roles is a Herculean task. But our committed postdoc volunteers have been hard at work designing this year's ISFS program. Our goal has been first, to inspire the next generation of future faculty and scientific leaders, and second, to celebrate and highlight the fantastic research of our selected fellows and associates.
This year's program intentionally takes us all through the transition from postdoc to faculty. Today, day 1 focuses on applying for and successfully acquiring a faculty position. We've kicked off the symposium with a very engaging address from Dr. Freeman Hrabowski III, and we also heard from a panel of assistant professors today about strategies to ace the job market. Looking forward, day 2 focuses on what it's actually like to be a PI, and we wrap up the symposium on day 3 with insights on how to flourish in a faculty position. Throughout, we will also continue to hear from each of our fellows and associates about their research.
We want to give a really big thank you to all of our postdoc volunteers and acknowledge them for their time, input, and energy in shaping this program. Together as a group, we have meticulously coordinated the topics, speakers, and moderators for each of these sessions over these 3 days to make it an enriching experience for all the attendees. We really could not have done it without everyone and all the efforts.
We also thank all of our invited speakers for joining us so enthusiastically and helping us to further the ISFS missions and goals. And finally, thank you to all of you for joining us today. We hope to empower you to participate in the sessions, ask questions, and engage with our speakers and content throughout this event.
We will end the day today with our first workshop of the symposium by Dr. Jennifer Kong. on organizing your job search process, specifically designed for our selected fellows and associates, and is also open to all our postdoc volunteers. Thank you all.
Thank you.